All right. So we've now covered the hepatitis, and what we want to do is start looking at the family as a whole of DNA viruses. So what we're going to do is go through some of the different virus families and look at some of the exceptions to the rule, okay? And then we're going to start by talking about the different DNA viruses. Okay. In the table, you'll see in your book, and I only have part of the table up here, or else you'd be squinting really hard to see it. You have all of the DNA viruses listed, right? The table tells you the type of DNA, does it have a polymerase, is it naked, enveloped, and whether or not replication occurs in the nucleus. You probably do not have to know specifically, is it single-stranded DNA, is it double-stranded DNA? Virion-associated polymerase for DNA viruses, probably not super important. Does it have an envelope or not? Yes, you definitely want to know that one. Where it replicates, unless it's pox, it's going to be in the nucleus, so that's fairly straightforward. And then what you certainly want to know, and what this table is really helpful with, is telling you the major viruses that are in each virus family. Why they like to ask these questions is they'll give you a little vignette of, let's say, slap cheek fever or B19. And then they'll ask you which virus family the agent causing the above illness is in. So you're very familiar with B19, but you're going to have to know also that it's a parvovirus. So definitely pay attention to the families that each of these viruses are in. Now, all of the viruses are double-stranded, really, except for parvo, so that one's not too difficult. But again, they're really kind of moving away from asking you specific questions about the type of nucleic acid. Remember, we're more and more clinically relevant now. We're moving towards clinical vignettes, so they're going to have to ask these questions in a clinically relevant manner. So as far as the DNA type, you're not going to stress out too much about that. How it replicates, where it replicates, is it enveloped? That's all very important. Now, all of the shapes of the DNA viruses are icosahedral. So that looks like kind of the little spaceship type looking virion I showed you earlier. And the only one that isn't icosahedral is pox virus. Pox virus, to be honest, we have no idea what the shape of that is. So we're very smart here. We call it complex, all right? So we don't necessarily know what it is. It doesn't fit into any of our categories. It's not helical. It's not icosahedral. It's complex, okay? And again, all of the DNA viruses are going to replicate their DNA in the nucleus except for pox virus. And we'll explain that a little bit more when we talk about the pox viruses specifically. So again, familiarize yourself with the different viruses in each family. Again, going down the table, adenovirus, one's pretty easy. Hepadnavirus, HEPA, meaning liver, DNA virus, okay? Herpes viruses, huge family of viruses. So you have to know HSV, VZV, EBV, CMV, and then human herpes virus 6 and 8 additionally, okay? Pox virus, you definitely want to know clearly smallpox, and then understand that molluscum contagiosum, which we do see infections with in the U.S., is also in that family, okay? So all of that information is nice and tidy for you in that table. Now what we want to do is look specifically at DNA viral replication using the herpes viruses as an example, okay? As we mentioned before, all viruses are going to have to attach, and the herpes viruses are icosahedral enveloped viruses. So they're going to attach to the host cell, and because they're enveloped and they have a fusion protein, their envelope can fuse back with the cell membrane and dump the virion into the cytoplasm. Now, remember we're talking DNA virus here, so the DNA has to replicate in the nucleus. All right, so we have to get uncoating of the genome, which we do, and the genome has to make its way into the nucleus for replication. So initially, we are going to get messenger RNAs, so we're going to get transcription from the DNA strand, and we're going to move back out into the cytoplasm so our ribosomes can hop on and we can start getting some early protein synthesis. All right? That early protein synthesis and intermediate protein synthesis that's going on are all making enzymes and proteins that are helpful for the virus to replicate. For example, the thymidine kinase that herpes virus makes that helps it with viral replication. Okay? Now, again, what's going to happen is we're going to continue getting protein synthesis, as you can see in kind of the bottom half of this slide. But now what's happening, if you go back and look in the nucleus, is we're starting to get some DNA made, which means we're having some genome replication, probably with the help of some of the proteins that we just made early on in that infection. Now we're going to move to the last stage of viral infection with the herpes viruses, where we're still getting genome produced, we're still getting some late protein synthesis, and remember what we said for late. We said late protein synthesis is going to go into making the structural component of the virus. So, herpes virus is actually a little bit unique in the fact that it actually assembles in the nucleus. Most viral agents will assemble in the cytoplasm and then leave from the host cell. Herpes viruses actually assemble in the nucleus. So the structural proteins that are made in the cytoplasm have to find their way back into the nucleus, and they will begin to assemble. The genome will start to associate with that icosahedron. It will then bud through the nuclear membrane, as you can start to see here. And here's the complete budding through the nuclear membrane. What you can see, though, is those studded lollipops, which are viral-encoded glycoproteins, essential for the virus to go and attach to another cell. Okay? So this virus is going to move through probably the Golgi, get any modifications it might need, and then it's going to do one of two things. It's going to leave the cell, as shown in this pathway, by exocytosis. Okay, so it's not going to blow up the cell. It's just going to leave the cell by exocytosis. Or, as we talked about before, that's really important for herpes viruses, is because of the fusion protein that herpes viruses have in the envelope, it can actually leave one cell and fuse another cell to the original cell, and now you get the formation of syncytia, or giant multinucleated cells. Okay, so remember, giant multinucleated cell is the same thing as a syncytia, and that's actually going to be helpful for diagnosis of some of the different herpes viral infections.